Welcome to the Deep Dive. Our mission today is to serve as your ultimate shortcut to becoming thoroughly well-informed about the hidden world of pathogenic fungi.
It's a vast world.
It really is. We are basing this on a highly curated 14-slide clinical atlas that covers the absolute foundations of clinical mycology. We're going to build this knowledge step by step, starting from their fundamental, almost alien biology, all the way up to how they execute these complex, deep tissue invasions.
Which gets pretty intense.
Oh, it does. If you ever wondered what's actually happening when a fungal infection takes hold, well, you are in the right place. To kick this off, we have to establish the ground rules. What exactly makes a fungus so different from a plant or a bacterium?
Well, the most crucial distinction is how they get their energy. Even though they were historically studied alongside plants, fungi are totally devoid of chlorophyll.
So, no photosynthesis at all.
None. They cannot convert sunlight into food. They are heterotrophic, meaning they require organic carbon to survive. In plain terms, they have to eat other things. They are the great decomposers. And the physical structures they use to do this are incredibly unique. The entire body of the fungus, it's known as the thallus.
Okay.
As it grows, it forms this large vegetative mat called a mycelium. And if you zoom in on that mycelium under a microscope, you'll see it's made up of these microscopic intertwined tubes called hyphae.
Okay, let's unpack this because the materials they use to build those tiny tubes are just wild.
Oh, yeah.
Their cell walls are largely composed of chitin and cellulose. And chitin is the exact same ultra-tough material that makes up the exoskeletons of beetles and crabs.
It's essentially insect armor.
Right. And if you put these fungal walls into a hypertonic solution to digest them, you get these bare cellular units called protoplasts. Plus, unlike bacteria, their cell membranes uniquely contain steroids.
Which is huge. If we connect this to the bigger picture, these structural choices dictate the entire field of clinical mycology.
How so?
Well, why doesn't penicillin cure a fungal infection?
Because it's the wrong tool.
Exactly. Bacterial antibiotics, like penicillin, are engineered to destroy a specific chemical in bacterial cell walls called peptidoglycan. Fungi don't use peptidoglycan, they use chitin.
Ah, so it's like trying to use a computer virus to hack a typewriter.
That's a great way to put it. You are firing the wrong weapon at the wrong target. Conversely, fungi are highly susceptible to specific antifungal drugs, like grizafulvin, which bacteria completely ignore.
I love that typewriter analogy. And their environmental preferences are totally different too, right?
Completely.
They are like the picky, sweet-toothed eaters of the microbial world. They demand high sugar concentrations to thrive, around 4 to 5%, and they prefer heavily acidic environments with an optimum pH between 4.6 and 6.0.
While bacteria, meanwhile, want a neutral pH and barely 0.5% of sugar.
Right.
This ecological niche partitioning is why fungi can thrive in places bacteria generally struggle, like highly acidic fruits or specific layers of human skin. But their structural makeup is just the beginning. The way they physically present themselves is where things get truly deceptive.
Right, because we know what they're made of. But what do they actually look like?
Yeah.
The atlas breaks them down into two broad shapes in clinical mycology: moulds and yeasts.
Yes.
Molds are those filamentous multicellular structures we just talked about, the big, fuzzy mycelial mats. Yeasts, on the other hand, are unicellular.
Just solitary little cells.
Exactly.
Yeah.
They reproduce by budding off one another.
Yeah.
They range from 1 to 5 micrometers up to maybe 30 micrometers.
Yeah.
And if you grow them in a lab dish, their colonies look deceptively like smooth bacterial colonies.
They really do.
But they don't just pick one shape and stay in that lane forever, do they?
They certainly do not. And this brings us to one of the most elegant and honestly terrifying mechanisms in microbiology: dimorphism.
The shape-shifting.
The ability to shift shapes, yes.
Yeah.
Many of the most dangerous human fungal pathogens exist as a mycelial mold when they are out in the environment at room temperature, which is around 25 degrees Celsius. But the moment they infect human tissue and hit our core body temperature of 37 degrees Celsius, they undergo a radical biological transformation into a yeast phase.
So wait, they basically wear a lightweight summer tee when they're lounging in the lab, but bundle up in a heavy winter coat the second they enter the human body.
That is exactly what they do. And it's an incredibly effective survival strategy. Think about the mechanics of it. Growing as a fuzzy mold in the environment allows them to produce and launch spores over vast distances in the open air.
Right, they need to spread.
But if you inhale one of those spores, acting as a multicellular mold inside your bloodstream would make them an easy, bulky target for your white blood cells.
They get wiped out.
Precisely. By rapidly shifting into tiny single-celled yeasts, they can navigate the bloodstream, multiply quickly, and effectively evade immune defenses that were expecting a totally different type of threat.
That is brilliant and terrifying.
Pathogens like Candid albicans, Coccidioides amidus, Histoplasma capsulatum, and Blastomyces dermatitis all use this thermal trigger to invade us.
There is also one major player called out in the atlas that uses a different kind of defense, right? Cryptococcus neoformans.
Ah, yes.
It is the only one in its class that is heavily capsated. It wears this thick outer armor, which we will see makes it devastatingly effective at crossing barriers in the human body.
It's a huge clinical problem.
So if these organisms are constantly shape-shifting based on temperature, how do scientists even begin to classify what they're looking at under a microscope?
Well, it requires an incredibly rigid system. Taxonomy here is governed strictly by the Botanical Code of Nomenclature.
Botanical, even though they aren't plants.
Right. Under Whitaker's five-kingdom concept, they sit in the kingdom myceti. But for human medicine, almost all the pathogens that infect us and our animals belong to one specific division, the Amastigomycota.
There is one subgroup within that division that really caught my eye, though. The Deuteromycetes, also known as fungi imperfecti.
Yes, the imperfect fungi.
They are largely pathogenic, but what makes them imperfect is that they completely lack sexual reproduction. They are just out there endlessly cloning themselves.
Well, the term imperfect is actually a reflection of our own scientific blind spots.
Oh, really?
Yeah, it wasn't that the fungi were broken, it was that mycologists hadn't yet observed their perfect or sexual reproductive stages in a laboratory setting.
Oh, I see.
But regardless of how they reproduce, their microscopic anatomical blueprints are stunning.
Here's where it gets really interesting. When you look at the major moles under a microscope, they look like bizarre alien vegetation.
I do.
Take the Phycomycetes, which include genera like Rhizopus and Mucor. These are non-septate, meaning their tubes don't have walls dividing the cells.
Right, they are coenocytic.
Essentially one giant, continuous microscopic hose filled with hundreds of nuclei sharing the exact same cytoplasm.
It's a very unique architecture.
And Rhizopus anchors itself with these root-like structures called rhizoids connected by stolons, while Mucor operates without those anchors entirely.
Contrast that with the Ascomycetes, which feature famous genera like Aspergillus and Penicillium. Their architecture is vastly more structured.
It's a night and day difference. Aspergillus looks like a bulbous dandelion head.
Very bulbous.
It has this unbranched stalk that swells into a vesicle at the top, and that vesicle is covered in little pegs called sterigmata that shoot out spores.
Right.
But Penicillium, the fungus that naturally produces the penicillin antibiotic, it looks completely different. It has branched septate stalks. It literally looks like a microscopic skeletal tree with multiple branches holding up chains of spores.
And every single one of those complex, tree-like microscopic structures exists to execute their ultimate biological imperative.
Reproduction.
Exactly. The sheer variety of ways they multiply is a testament to their evolutionary resilience.
Let's ground this for the listener, though. If you are sweeping out your dusty garage and accidentally kick up a cloud of debris, what are you actually inhaling? How do these structures guarantee there are always spores waiting for us?
What's fascinating here is the sheer mechanical diversity of their spores. On the asexual cloning side, you have zoospores, which are actually motile and can swim in water.
Wow.
You have aplanospores, which are non-motile. You have conidiospores, which are literally pinched off the sides of the hyphal walls like tiny buds.
And sporangiospores that grow in a sac, right?
Yes, and thalasspores that form directly from the original hypha.
But the ones that blow my mind from the atlas are the survivalists, like the chlamydospores.
Those are top.
They don't just pinch off a spore and hope for the best. When conditions get harsh, maybe the environment is drying up or the temperature drops, the fungus concentrates its protoplasm and builds a massively thick wall around it.
A literal doomsday bunker.
It essentially creates a bunker,
Yeah.
or arthrospores, where the hyphae themselves literally fragment and shatter into oblong infectious pieces. Then there are blastospores that just bud right off.
And that's just asexual reproduction. When they reproduce sexually, which allows them to mix genetics and adapt to new threats, they utilize male cells called the antheridium and female cells called the ocogonium.
Right.
If two similar gametes fuse together, they create zygospores, which is common in Rhizopus. But if two totally different cells fuse, they produce eucospores.
The packaging for these sexual spores is wildly precise, too. Ascospores develop endogenously, meaning internally, and they are packed exactly 8 to a microscopic sac.
Exactly 8.
Meanwhile, Bethidiospores develop exogenously on the outside, sitting exactly 4 to a club-like structure.
The mathematical precision is almost unsettling.
It really is. But with all these invisible, highly protected spores floating around, it raises a huge practical question. When a patient comes into a hospital incredibly sick, how does a clinical lab actually isolate and identify these shapeshifters?
Well, it requires immense patience in very specific chemical environments. Cultivating fungi is nothing like bacteriology. A bacterial culture might give you answers overnight, but pathogenic fungi are slow, deliberate growers. Labs use media like sabro-dextrose agar, or SDA, and blood agar, and they have to incubate them at temperatures between 30 and 37 degrees Celsius for 1 to 2 full weeks.
A week or two. That is an agonizing wait when a patient is declining.
It is.
And I was looking at the chemical tools they use to isolate the fungi during that stand wait, and it sounds like a nightmare. They use inhibitors to filter out the background noise of harmless environmental molds so they can find the pathogen.
That's right.
But one of the main inhibitors they use, cyclohexamide, is actively toxic to Candida species and Cryptococcus neoformans. Wait, we use cyclohexamide to filter out the junk, but it accidentally kills some of the exact dangerous yeasts we might be looking for? That sounds like a diagnostic tightrope.
It is the ultimate diagnostic tightrope. Think of it like trying to weed a garden where the invasive weeds and your prize-winning roses are biologically identical. The poison you use to kill the environmental junk is the exact same poison that will kill the deadly pathogen you are desperately trying to find.
So what do they do?
If a lab technician suspects Candida or Cryptococcus, they have to run parallel cultures without that inhibitor, knowing it will be overrun with background molds, just to ensure they don't get a false negative.
Which makes direct examination of the patient's tissue absolutely critical while you're waiting those 2 weeks.
Essential.
Taking skin or hair scrapings, treating them with a 10% potassium hydroxide or KOH solution, and applying a little heat dissolves the human tissue so the tough Keaton-walled fungi remain visible.
Yes, the KOH prep.
They even use a Woods lamp, which is a UV light, to shine on a patient's scalp because certain fungal hair infections will actively fluoresce in the dark.
And once they finally isolate a sample on a glass slide, the clinical stains they use are fascinating.
Yeah?
Lactophenol cotton blue is the standard for a rapid mount to see the structure, but the PAS stain is brilliant chemistry. It specifically reacts with the unique carbohydrates in the fungal cell wall, turning the fungus an intense blazing red or magenta color, so it stands out starkly against the pinkish background of human tissue.
Okay, so once the lab stains it magenta and identifies exactly what we're dealing with, what is that fungus actually doing to us? How does the disease unfold?
Well, first we have to differentiate between a fungal attack and a fungal poisoning.
Right.
Mycetism refers to the toxic effects of directly eating poisonous mushrooms, which causes severe biological fallout.
Right.
Mycotoxicosis is when a disease is caused by fungal toxins leaching into our environment or food supply. A notorious example from the atlas is aflatoxin, a highly dangerous liver carcinogen produced by the mold Aspergillus flavus.
But when we look at actual infections, the invaders that set up shop inside us, we usually start with superficial mycosis.
The skin eaters.
Right, these are transmitted by direct contact. We call them dermatophytes, and they cause ringworm or tinea. They can attack the scalp as tinea capitis, the nails as tinea unguium, or the feet as tinea pepitis.
What is truly remarkable about dermatophytes is how specialized they have become based on their evolutionary habitats. The clinical diagnostic matrix categorizes them perfectly.
Yeah, the habitats are wild.
They are anthropophilic, meaning they have evolved to specifically target human hosts.
Or zoophilic, targeting animals but occasionally jumping to us.
Or geophilic, meaning they live in the soil and are opportunistic, and their structural shape tells you their limitations.
And their physical characteristics dictate exactly what parts of your body they are allowed to attack. The genus Microsporum features these abundant fusiform macroconidia, and it targets hair and skin.
Right.
Trichophyton features abundant tiny microconidia, and it's a triple threat. It has the enzymatic tools to infect hair, skin, and nails.
It takes it all.
But epidermophyton, which has club-shaped macroconidia, crucially causes NO hair infection. It lacks the ability entirely. It sticks strictly to the skin and nails.
The biology dictates the pathology. If a clinician sees a patient with a severe fungal hair infection, they immediately know they can rule out epidermophyton.
That is so precise.
But the truly terrifying scenarios happen when fungi don't stay on the surface. When they breach our deep tissues or when they take advantage of an immune system that is already distracted, the results are devastating.
Let's look at the deepest, darkest part of this deep dive. Systemic and opportunistic mycoses.
Systemic mycoses are incredibly dangerous because their yeast phases allow them to mimic other severe diseases. Blastomycosis, for instance, clinically resembles severe lung tuberculosis, though the atlas notes it's interestingly absent in India.
Huh.
Then you have paracoccidioidomycosis. You have coccidioidomycosis, which
Mhm.
utilizes those fragmented arthrospores we discussed earlier to establish deep lung infections. And then you have histoplasmosis.
Histoplasmosis is pure nightmare fuel. When you look at the tissue samples, you don't just find the fungus floating in the blood. It is found inside the patient's own macrophages.
Yes.
It is literally hiding inside the exact white blood cells that are supposed to be destroying it. And its survivalist clematospores feature these creepy, unique, finger-like projections.
It uses our own cellular defenders as a Trojan horse.
Insane.
And then there is cryptococcosis, caused by Cryptococcus neoformans. Remember that thick capsular armor we mentioned?
Yeah, the only capsulated one.
That capsule allows it to cross the blood-brain barrier with terrifying efficiency, giving it a devastating affinity for the central nervous system.
Wow.
Beyond these systemic threats, we have opportunistic and invasive mycoses, fungi that are everywhere but seize the moment when we are weak. Candida causes severe thrush. Aspergillus fumigatus, characterized by its notably green colonies, invades the deep lungs.
And mucormycosis, caused by mucor and rhizopus, which aggressively invades blood vessels. So what does this all mean when these infections take hold? Look at this one human endemic infection highlighted in the slides, rhinospiridiosis.
That one is shocking.
It's endemic to South India and Ceylon, and it is caused by R.C. berry. It produces these massive polyps in the mucous membranes of the nose, eyes, and ears. And the sporangia, those tiny microscopic sacs inside the polyps, contain 16,000 spores.
16,000.
16,000 infectious payloads in a single microscopic sac.
The math behind that payload guarantees that the moment that sporangium ruptures, the pathogen's continued survival and spread is absolute. And we must remember, this isn't just a human war.
Yo.
There's a massive veterinary toll. Cattle suffer from mycotic abortion and severe mastitis. Equines are affected. Birds suffer massive crop inflammation.
It's everywhere.
These fungi can even bore into bone and deep structural tissue, causing conditions like meduromycosis and osteomyelitis.
It is a relentless global pressure across every mammalian and avian species. Which brings us to a fascinating historical problem. Because these pathogens are so adaptable, so good at hiding their true forms, human medical history has genuinely struggled just to keep track of what to call them.
Exactly. For decades, clinicians were documenting the severe symptoms in patients and naming the diseases without fully understanding the complex organisms causing them.
Right.
This led to a tangled web of historical nomenclature.
The atlas lists a bunch of these. Darling's disease is actually histoplasmosis. Amida disease is paracoccidioidomycosis. European blastomycosis is cryptococcosis. Valley fever is coccidioidomycosis. And Gilchrist disease is blastmycosis.
And remember the fungi imperfecti, the Deuteromycetes we thought only cloned themselves.
Yeah, the ones missing their sexual stage.
Well, as mycologists slowly improved their lab techniques over the decades, they finally managed to coax out and observe those hidden sexual reproductive stages. But doing so meant the entire botanical nomenclature had to be updated to reflect their true family trees.
Oh, wow.
Cryptococcus neoformans is now officially, botanically, Phyllobucidiella neoformans. Histoplasma capsulatum is now Agelomyces capsulata.
Even though doctors in the clinic still use the old names just to avoid confusion. And to bring this all brutally close to home, where are two of the most dangerous systemic pathogens hiding. High concentrations of both Cryptococcus and Histoplasma are found right above our heads, incubating perfectly in everyday pigeon droppings in our cities.
They are everywhere. Which brings us to a critical realization about the future of this biological war. This raises an important question.
All right.
We know that the most dangerous weapon these fungi have is dimorphism. Their ability to shift into a deadly invasive yeast phase the exact moment they hit our 37 degrees Celsius body temperature.
Right, the summer tea to the winter coat.
Exactly. Our mammalian body heat is essentially a thermal barrier that keeps most environmental fungi at bay. But as climate change accelerates and global environmental temperatures consistently rise, these fungi are being forced to adapt to hotter conditions just to survive in the soil.
Oh, no.
If environmental fungi adapt to thrive at higher heat, effectively bypassing our mammalian thermal barrier entirely, are we about to face a completely unprecedented wave of shape-shifting pathogens that are already pre-adapted to human body heat?
But that is an absolutely chilling thought to leave on. If the summer tea and the winter coat suddenly become the exact same temperature, our primary defense mechanism is just gone. We started this deep dive talking about how bacteria are like simple, predictable robots.
Yeah.
And they aren't.
But after exploring everything from their chitin armor and thermal shapeshifting to their massive spore payloads and ability to hide inside our own immune cells, it's clear we are facing a brilliant alien ecosystem, one that has been perfecting the art of survival long before we ever arrived. Thank you for joining us on this deep dive into the foundations of clinical mycology. Keep exploring, keep questioning, and we'll see you next time.